Hello, everyone, and welcome to our webinar today. Thanks so much for joining. It's a great pleasure to be here today with two amazing speakers. My name is Christoph, and I'm the Managing Director of Genomics. We specialize in 3D cell culture products, and some of our products are particularly based on gelatin-derived matrices. And it's a great pleasure and honor today to have two fantastic speakers who have a lot of wealth of experience in that particular field, and also the possibility for us to hear a little bit about their research and how they apply extracellular matrices like this in their applications. So we've got Dr. Koen Lim joining us today and Professor Travis Klein. And I guess without further ado, would like to introduce Dr. Koen Lim. He's a senior research fellow at the University of Otago in Christchurch, New Zealand. Koen holds a PhD in biomedical engineering from the University of New South Wales in Australia, and he specializes in hydrogel polymer chemistry, 3D bioprinting, and tissue engineering, particularly photocross-linkable materials. Koen, you've got a very impressive track record, over $4 million in funding, prestigious fellowships, and lots of awards, so really well done here. Published over 50 papers and also has a couple of patents, so fantastic work. And before I'll let you start your presentation, I just wanted to point out to all the attendees, please, if you have any questions, just use the chat function in the GoToWebinar control panel on the right-hand side. Also, please feel free to ask them during the presentation. And after Koen finishes this presentation, we'll go through the questions one by one and give him an opportunity to answer these. So without further ado, I'll just change the screen over to Koen, and I'm looking forward to seeing your presentation. Here we go. That works.
All right, and you can see my screen, yeah?
Yes, we can see it. Thanks.
Awesome. Thanks a lot, Christoph, for the invitation. And it's great to really, my huge pleasure to be able to present some of my work that we're doing here at the CREATE group in Christchurch. I'm part of the Department of Orthopedic Surgery and Musculoskeletal Medicine. So a lot of our work are looking into the joints, so the bone, vessels, cartilage, which I believe Travis will also talk a bit about it later. So my research focus is a lot on light-based materials or light cross-linkable materials. And for today's topic, I believe we're talking about gelatin-based hydrogels, and I quite like to put this slide up most of the time because gelatin is not foreign to a lot of people. The fact that we use gelatin a lot in cooking and in baking, and you get these really tasty cakes that you can make. But really, the reason why we're using it is because it's denatured collagen. It has all of these really good advantages that we would like to culture cells in them. So water solubility, abundance of self-responsive motifs. So we know that it still retain a lot of these RGD sequences to signal with cells. We know it's temperature sensitive, so it undergoes soil transition just depending on changing temperature. And also we can easily modify it. And for my research with looking at light cross-linkable materials, we often modify gelatin with some photo label moieties. So I always make a joke where none of our printed construct will be as pretty as any of these desserts or tasty. But I did find out that people are using 3D printing of gelatin in the food industry at the moment. So there's this company in Japan looking at 3D printing of sushi. So at some point you can make 3D printed tasty products, which is kind of interesting for it's very futuristic. But really what we're doing in our group is looking into multiple biofabrication platforms. We do extrusion-based printing, we do lithography-based printing. For the sake of today's talk, in response to time that we have, I'll really just be focusing on what we have been doing in the extrusion-based and some of our recent publications and some unpublished work. So we were very interested in doing photo cross-linking, and photo cross-linking has been applied in multiple different bioprinting technologies. And we've just published this review recently, last year actually, just 1 year from now, looking into the integration of photo cross-linking or photopolymerization into these different extrusion-based or lithography-based biofabrication technologies. And we did identify that one of the 4 of the important components to be look out for will be the photoinitiators, which I'll talk about, the photo irradiation conditions, the photo cross-linking reactions, but also the application. So what are you using this material for? And that does dictate how are you going to vary these different conditions and different parameters. So we've published this last year, so you can look this up, this publication, if you're interested to know more into the fundamentals of the chemistry behind it. But really, I'm interested in light because of its spatial temporal control. So if you have a photo polymerizable macromer or solution, and you project a pattern of light onto it, and you can very see that wherever it cross-links is where the light is being projected to. And you can also look into the resolution, and in this case, we were projecting a Mickey Mouse pattern onto this, trying to get it to really fine resolution. And when I slightly gently stroke, you can see that Mickey Mouse is sort of dancing here. So you're getting the resolution in comparison or in response to the light-based chemistry that you're using. So we do a lot of work on modifying gelatin. So one example that I've picked here is using gel-H. So we took gelatin, which is a gelatin backbone. We modify it with some allele groups, in the presence of a thiolated cross-linker and a photoinitiator. It undergoes the step growth thiolinic chemistry and it forms a hydrogel. The chemistry behind the bio-ink itself is kind of important, with the fact where if you look into the reaction between your macromer, your crosslinkers, and your photoinitiators in this instance. In our group, we use a visible light activated photoinitiator that is based on the transition metal complex, ruthenium. When ruthenium is excited in the visible light range of 400 to 450 nanometers, it gets into excited into ruthenium. It gets into the excited state, and then it donates an electron to a sodium persulfate. And we do know that these sulfate radicals can then react with these thiolated crosslinkers, for instance, in the system, and you can undergo a redox reaction. So when Brown, my PhD student, was looking into this system, we were looking into, can a primary redox reaction start imparting mechanical stability to some of these bioinks? So Brown looked into, a really soft formulation. So we're looking at a really low weight percentage, so just 3 weight percent gel-H with the ruthenium SPS photoinitiators and with PEG 8 arms as our crosslinkers. If you just mix it, because of its low viscosity and low weight percentage, it normally comes out in the puddle. But what we found is that in the response to time, because you have this primary redox crosslinking happening, that is a reaction between the persulfate and the thiolated crosslinkers, you do see a sol-gel transition happening just in response to time. And what this really means that if you mix your component into this syringe and you let it sit there for a while, you slowly get some shape fidelity. So from 0 minutes to 15 minutes, you go from a puddle to some sort of a gel-like shape. So that's just primary cross-linking. If you give it more time, you have an increase in viscosity and it's get better to extrude it. So I really wanted to show this as a way that we're using the chemistry of the basic macromer in coupling with the photoinitiating chemistry to impart some sort of mechanical stability to extrude these soft bioinks. We also looked into, if you extrude it and then incubate it directly without the secondary or without exposing it to light, the whole gel will dissolve. And we do found that you do need to irradiate with light in order to give its maintenance of shape stability over time. We were worried that if you incubate it for so long, 90 minutes with cells in there, how would that actually impair the cell viability? When we look at putting our MSCs in them, we don't see any differences in terms of the viability through the prints or with just direct cross-linking. So if you just mix them all and just cross-link with light. And when you look at the morphology of these MSCs, you can see that they can undertake this native spread morphology, which is sort of what we are after in these soft bioinks. So this is some work that we have done and published last year on stepwise control of low density bioinks for printability. And really moving forward to that, and I guess what I really wanted to highlight more is the potential of these visible light photoinitiators. So one work that I've done with Dr. Ricardo Lobato here from University of Utrecht is looking at the integration of, if we make these printed constructs and we want to integrate it back to the native cartilage, how do we get binding between the native cartilage and the bioprinted construct at the interface. So we look into taking JELMA. JELMA is a very well-known gelatin-based bio ink, which we then further functionalize it with tyramine. And I'll talk about in a minute to why we're using tyramine to get JELMA tyramine. And with the VisiBlight initiators, we do know that it is possible to facilitate this dityrosine cross-linking. So we're hoping to get enhanced integration with the native cartilage through the biphenyl bonds. And this is just giving a little bit of a background of when I was talking about GelH earlier, I was looking at the sulfate radicals, but now we're talking about this dityrosine or biphenyl bonds that's happening. It's now looking at the different activated products during this photo cross-linking process. We're looking at the highly oxidized ruthenium 3+, which then facilitates this kind of cross-linking here. So what we really found with this is that if you use JELMA tyramine and with the VisiBlight photo initiators, you get much better adhesion or binding between the hydrogel, so the JELMA tyramine, and the native cartilage. So there seems to be a 15-fold increment in the adhesion, and that is due to the dityrosine that we hypothesize is happening. And you can really see one of these really nice examples here, where you can take two pieces of cartilage, glue them together, and you can hold it and walk around the lab and it doesn't fall off. So this is just to showcase one of the extra functionality of these VisiBlight initiators, that it doesn't only just facilitate unsaturated ester bond or radical-based cross-linking, but it can also facilitate native dityrosine cross-linking, which most proteins would have the native tyrosine moieties. We're able to print this as well. Another thing that I would quickly want to talk about is more on sacrificial 3D printing. So we've done some work with silk, and I won't talk too much about it today because our topic is on gelatin. But one way to use extrusion-based bioprinting is to print this sacrificial template. In this case, we were printing pluronics. You can make them into different scaffolds, different architecture, and then you can extrude a low-density bio ink within them, and then cross-link this to impart some sort of structure into these constructs. What we showed through this work with Jelena is that we are able to control the strand thickness, the designs of pluronics. Pluronics prints really well. I'm sure all of you that has worked with pluronics know that is one of the gold standard that we use in terms of optimizing the printing. You can control the different orientations as well, and that gives us this kind of functionality or this kind of flexibility in making constructs of thick fibers, thin fibers, big channels, small channels, all in one, and using sacrificial-based bioprinting. And this is a really nice figure that I thought that I would like to show here, is that you're able to then get 3 different fibers of 3 different diameters, so 40, 75 microns, and 120 microns, where you can stack them all in one scaffold. So we can have some sort of a gradient within this scaffold, and we can also start controlling the fiber spacing, so how far they can go, so 100, 250 micron to 400 microns, and we know we can print them. We do have a follow-up work here that we showed how these architectures affect host tissue integration. We've published that earlier, late last year as well, sorry. But what I really wanted to show how this is then applied to gelatin is because we know that gelatin has some native tyrosine. So if we use this VisiBlight photo initiators, you're able to also then cross-link this gelatin-based hydrogels. So Bram, again, my PhD student, was looking into now using gelatin cross-link with these VisiBlight initiators as sacrificial bioink. So instead of using pluronics, which we know would just instantly dissolve once you print it and then you try to wash it away, can we use sacrificial bioinks that we can sort of control the degradation time frame? And Bram did a comprehensive study, and he systematically looked at the different weight percentages, the different photo initiator concentrations. And it does seem that we can then tune the degradation of these gelatin bioinks. And we're now then looking to using them for sacrificial base as a replacement for pluronics. So if you just print pluronics into these hydrogels, you wash them, you get instant dissolution, so you open up these channels instantly. If you use this delayed dissolution of gelatin-based bioinks, you can see where after 1 day you still get this whole bulk gel. So we have gelatin sacrificial bioinks embedded within another hydrogel, but over time, which we can control, these channels will then start to open, We know that we can print this sacrificial gelatin-based bioinks into different layers, different structures with different polymer concentration. But what's really important that we found is that there is a difference in terms of instant dissolution versus delayed dissolution. And we really just think it's the change in the microenvironment of the hydrogel itself that probably goes from hypoxic to a normoxic condition. So when we put MSCs in them and try to direct them down the bone differentiation, osteogenic lineage, you do see more mineralization in the delayed dissolution samples, but when we also look at vascular formation, we also see much longer and much more mature microcapillary network in the delayed dissolution formulations here. So this is some ongoing work that we're still trying to write this up for a publication. So I think with that, probably up at my time now. So it's a quick snapshot of the work that we're doing here at the CREATE Group in University of Otago here in the Christchurch campus. We look at different light activated materials. Today I've just really showed more around the extrusion-based bioprinting. We do some other work as well in terms of growth factor delivery or adhesives, but that is for the next time. So I am editing a special issue in the Journal of Biofabrication on harnessing light in biofabrication. If anyone is interested to contribute, please get in touch. Happy to look at any pre-submission inquiry. And I would just really like to acknowledge the team here. So my long-term collaborator and mentor, so Tim Woodfield, which probably not foreign to a lot of you, but also the PhD students that worked hard in generating some of the data here. My current team, and also the different funding and collaborators that I have. So thank you very much, and I'm happy to take any questions.
Thank you, Koen, for this fantastic presentation. Some really, really nice data and well presented as well. So just for all the attendees, if you have any questions, feel free to just type them into the chat or question function and go to webinar, and Koen will be happy to answer these. Maybe I'll just start off with a couple of questions. I was really intrigued by your data, really beautiful work there. I had a question concerning the thiolene click chemistry, step growth, gelatin-based gels. So do you see that these type of gels, because they are step growth mechanism, are they more stretchy compared to a Gelma gel? What about, you know, the ultimate strength if you are looking at, let's say, at the toughness of the materials? Do you see a major difference there because the chemistry is different?
We saw a difference in durability. So when we compared GelH to Gelma, so the step growth thiolene hydrogels are definitely a lot more durable in the sense that we're able to even fold it if you make it into a really thin film, as compared to when you do Gelma and try to fold it, it normally just snaps because it's kind of brittle. But the real reason I think that I'm interested in this kind of chemistry is the ability to control the microenvironment through just changing the crosslinkers. So some of the work I showed today is using DTT, which is a linear dual thiolated molecule. So it has two thiols at the end, and it would then just clicks. But you can also do polyethylene glycol with 4 arms, for example, or polyethylene glycol with 8 arms with different thiols on it. You can also control the molecular weights of this crosslinker, and that just gives you a lot more flexibility to the final printed construct that we have.
Fantastic, thanks for that. Maybe I'll just ask one more question. We have a few questions also from the audience, but I'll just have one more that I find quite interesting. So you also showed that with this type of chemistry, you can basically pre-crosslink the material, which allows you then to better extrude it. So you get a better, I guess, shear thinning behavior and a shape fidelity after printing. How much of this behavior is attributed to the natural property of gelatin to thermally gelate if you have it at room temperature versus the formation of any type of crosslinks through that chemistry?
Yeah, well, I think the key there, Christoph, always is, when we're trying to look at the rheological profile, the key there is find the balance, right? You either want something that doesn't change too quickly, so you get more control, but then you still want it to be fairly reactive once you give it a stimulus so that it maintains its shape. The work that we publish here, we really look into low density bioinks. So we're looking at really low weight percentages. So we're looking at 3 weight percentage gelatin, for example, where without the photoinitiators and without the thiolated crosslinkers, they don't set. So we don't see that setting behavior because they're just really low. And that gives us a much better control over the temperature that we can print it, we think. Yeah, you know, there's different kinds of gelatins. Of course, we didn't look into different types of gelatin. We were looking at just type A, porcine, you know, the standard one everyone is using. But assume if you use type B, you use bovine, you use fish gelatin, even though everything will change again. So it's a whole new optimization process.
Absolutely. Thanks for that answer. So we've just got a question here from the audience. The question is, what is the in vivo degradation profile of GelMA, let's say 80% degree of functionalization? Can you maybe comment on that from your experience?
We have studied long, well, I said long-term degradation, but it's really just about 3 months in a subcut model using a 60% degree of functionalized gelatin. And at the time of harvesting the samples, the gels were still beautifully intact. So it doesn't seem to be great. So I guess the question is, you know, the in vivo degradation will be depending on where you put it. If you put it in a highly dynamic environment, it will be different. If you put it in a place where you don't get a lot of fluid that goes through it, it will be different. If you put it into a wound healing site where you have a lot of MMPs, a lot of proteases that is remodeling the surface, then the gel would degrade much faster. Bear in mind that gelatin itself, you know, is susceptible to enzymatic degradation. I don't know. Did you lose me?
That's exactly right. From my experience as well, I would definitely second that. Say, for example, sorry, just had some connection issues there.
Yeah. I don't know. Did you lose me? Am I back?
Okay. Well, just one last question, maybe from my side. Oh, yeah, you are back. It's a little bit bumpy connection there, but we can hear you now. So if you have anything else to say, please feel free.
Oh no, I think I
Alternatively, I have another question as well.
Yeah.
Yeah. Perfect. So if you don't mind, I'll just do one last question, which is concerning the sacrificial bioink. I mean, this is a fantastic concept. I really like it. I can see, you know, millions of applications where you want to have the sacrificial bioink there for a little bit longer and then essentially degrade it over time. So that whole idea of being able to control the degradation profiles and really, you know, promote tissue growth and integration is a fantastic idea here. And in this context, I was curious, what has been used as the actual matrix to carry the cells? Is it this? It's not the sacrificial material, right? It's the second bioink that was used.
Yeah. So we looked at a number. So we looked at GelMA, we looked at gelate, we looked at silk, and depending. So I think I like the concept of the sacrificial bioink because you can then control the mechanics of the bulk gel. So as for example, we know that GelMA is a lot. You can get much higher stiffness than if you do a soft or just a gelatin-based hydrogel. So that gives you more of a flexibility to tailor it to the specific application that we're looking at. And you can then also, in the bulk hydrogel, for example, look at different materials. So we look at synthetic material, so like but silk, for example, which doesn't have RGD sequences. But we also can then look into gelage of different mechanical stiffness and different cells that you want to put in there.
Fantastic. Well, thank you so much, Kun, and thanks again for your presentation. Really some amazing work, and all the best for your future research. I think you're doing a fantastic job and going a great direction as well. Okay, so with that, I'd like to just ask Travis if you could just turn on your camera so we can see you as well. Here we go. So it's my pleasure to introduce you, Professor Travis Klein. Travis and I go way back. Travis actually has been my PhD supervisor, and I think I first met Travis in 2010, I think it was. And since then, we've been working very closely together, and you know, I've been on quite a few journeys together as well. So thanks for that, Travis. Travis is a professor here at the Queensland University of Technology. He's the director of the Center for Biomedical Engineering at QUT. He's also the leader of the cartilage regeneration laboratory at Queensland University of Technology in Brisbane, Australia. and he's also co-founder of Gelomics. So it's very exciting to have him here on board as well today. Travis holds a PhD from the University of California in San Diego in the US, and his expertise is really in tissue engineering, particularly around cartilage tissue engineering, also functional tissue engineering, biomaterials, and hydrogels, of course, as well. Travis has had a fantastic track record with some really nice, prestigious fellowships, and obviously also very good university to do a PhD, and over 100 published articles. So very excited to hear some of your latest research today, and yeah, we look forward to your presentation. So I'm just going to share your screen.
Okay.
Mr. Presenter, here we go.
Show my screen. Does that come through?
Yes, it just came through now. Thank you.
Okay, cool. Well, thanks a lot, Christoph, for the invite and for the introduction. It's a pleasure to be here, and also thanks to Koen for that really nice talk on gelatin-based hydrogels to start off. I'm going to be talking a bit about a particular application for gelatin-based hydrogels and GELMA in particular. So that's in cartilage tissue engineering, and tell you a bit about where we've gone over the past several years. Let me control my slides. Yeah, as Christoph said, I'm one of the founders of Gelomics, and this company really started with the four of us, Christoph and Pete, who were PhD students of mine in Dietmar's. We saw GELMA as a great material, in particular for cell culture, and we saw a need for this, and founded Gelomics in 2018. And a lot of it is based on the work that both Pete and Christoph had done during their PhDs. So GELMA, Koen has already introduced you to in his talk, but it really, I think it is really quite a versatile material that can be used in many applications based on gelatin. So gelatin already comes with a lot of things like matrix degradation sites, as well as cell binding sites. Cells can be easily encapsulated inside the GELMA, and you can give that three-dimensional matrix, which is critical for physiological sort of cell cultures. It's also quite tunable in terms of mechanical properties, and we know that mechanical properties are quite important in terms of influencing the cells. So GELMA is really just a modification of gelatin with these methacrylate or methacrylamide groups. We've looked in detail at where these functionalization take place in terms of which amino acids. You know, typically people look at NMR and see, okay, there's these two peaks here. It indicates functionalization, and you can quantify the degree of functionalization. But we've taken the approach of using mass spec and looking at each individual amino acid and where you actually have the functionalization. So it turns out that lysine is where most of the functionalization is happening. So you're mostly getting methacrylamides in the typical GELMA preparation, but you do also have functionalization on other amino acids. As I said, it is easy to tune GELMA properties in terms of the mechanical properties. You can change the concentration, and you can get gels that are, you know, a few kilopascals in stiffness, or increase the concentration and get 150, 200 kilopascal sort of stiffness. Another way to do it is to change the light exposure when you're photocross linking. You know, short exposure, you can get soft gels even with 20% GELMA in this case, or if you expose longer, you get stiffer gels. So that's good if you want to affect cells to form different types of tissues. We know that different cells are in environments with different mechanical stiffness, and so if you really want to direct either stem cells or other types of cells to form the appropriate tissue, getting that ECM stiffness is very important. And so if you want to make a model where you're forming blood vessels, for example, you want a soft gel. For cartilage applications, it's normally a relatively stiff matrix that chondrocytes are in, and so we've gone for stiffer gels, typically in the tens of kilopascal sort of range, which is still lower than the normal stiffness of cartilage, but it allows chondrocytes to form new tissue. So yeah, in cartilage, it's a very demanding environment. You have regular joint loading being applied to the cartilage at the ends of bones, such as in the knee. Cartilage can last, for many decades under these harsh conditions, and that's due to its low friction surface and the ability to bear load without wearing. But it does have the problem that when it's injured, it doesn't repair well. So this is just a histological section of cartilage showing that it's really mostly extracellular matrix and water. You have cells that are sparsely populated within that matrix, and they're responsible for maintaining that matrix. But when you get a cartilage injury, that doesn't have a good repair response. There are no blood vessels, no nerves within articular cartilage, and what happens over time is that you get degeneration of the joint and osteoarthritis, and the cartilage might end up looking something like this. Tissue engineering offers the potential to repair and replace that damaged cartilage, and thereby avoid development of osteoarthritis. So we've been looking at Gelma-based hydrogels as a potential matrix for cartilage tissue engineering, and we want to know a few different things about whether or not this material is suitable. So first of all, do cells survive well in that hydrogel? And do they behave as you would like? Do they make collagen type 2? Do they make aggrecan? Do they increase their mechanical stiffness with time as they generate that matrix? So if we look at just cell-free Gelma, this is an example just to look at degradation in vitro. We see Gelma basically is maintaining its compressive properties over time, up to 8 weeks in culture. In this experiment, we were looking at combining Gelma with small amounts of hyaluronic acid or chondroitin sulfate or both, and seeing if that has any effect. You can see for the cell-free gels, it doesn't make much of a difference. But when we encapsulate chondrocytes, we see that Gelma alone, we do see an increase over the first 5 weeks of culture in mechanical stiffness. But when we add hyaluronic acid, this is only at 0.5 weight percent to the Gelma, we end up with a major increase in the compressive modulus, which indicates that it's forming a functional matrix that is developing over time. This is just showing what it looks like. We could look at different concentrations of hyaluronic acid, methacrylate or HAMA in here, and looking at the aggrecan, we see more diffuse staining away from the cells, and that is what is providing that mechanical advantage. So, like I said, in cartilage, it is constantly loaded as you walk around, as you jump, lift things. Your cartilage is being loaded. It is important if you're developing a tissue engineered construct for cartilage applications that it's able to respond to load. So we've been studying the effects of compression and shear on constructs. including these Gelma-based constructs in a custom bioreactor. This is one that Christoph developed during his PhD. And we looked at things like compression. How does that affect the gene expression? So we can see dynamic compression increases agrochem, increases collagen type 2 expression, and also proteoglycan 4, which is the lubricant that's found and maintains that low friction surface. We also have done similar things with shear loading, and can see at high levels of sliding, we can see increases in proteoglycan 4 as well. And then the combination of the two, where you see, again, a good effects in terms of chondrogenesis and production of proteoglycan 4. If we look at immunohistochemistry or immunofluorescence level, we can see actually more accumulation of important molecules like collagen type 2 when we're loading them. So the mechanical loading is affecting the cells, and it's with the right loading protocols, you can improve the production of cartilage like ECM. If we look over the entire construct here, you can see collagen type 2 throughout. You can also see collagen type 1 largely at the periphery, and this is an issue that you have in vitro when you culture cells in a three-dimensional environment. You end up getting cells growing out on the surface and forming a layer which has a lot of collagen type 1. When we loaded these constructs, we actually didn't have that effect on the surface. We didn't have that overgrowth in collagen type 1 on the surface. So speaking of the surface properties, that's an area that I think a lot of people don't think too much about when they're doing cartilage tissue engineering, but it's very important in terms of the long-term function of the graft. So ideally, you want a very, very low friction surface and one that doesn't wear. So when we were making our gels, we typically make them in a mold. You can 3D print or use photolithography as Kuhn pointed out in the first presentation, but we typically make them in molds with Teflon being the main material for the mold, and then a glass surface. If you make gels like this, you will see that there are differences in frictional properties between the Teflon exposed side and the glass exposed side. And we studied this and saw that indeed the coefficient of friction is much lower in the Teflon exposed side, and friction coefficient much lower than 0.1 as well. It's dependent on the sliding velocity, but that is maintained over time in culture as well. And we can see with repeated loading, you still maintain this difference between the Teflon and the glass slide in terms of friction coefficient. We've looked into it in terms of how or why this is the case. And with AFM, we can see that the surface is actually more rough on the PTFE exposed side as compared to the glass. And using cross-linked gels and then probing it with a methacrylate functionalized dye, we can see that the dye is incorporated at the PTFE side much more than at the glass side. That indicates that the surface is not completely cross-linked, so you have these dangling chains at the surface. We've looked in terms of deformation as well. We've generated some little devices where we can mount on the microscope, and then using digital image correlation, investigate the strain in different regions. Here we have a bilayered construct where we have two different stiffnesses of gel, and you can see the see the varying colors indicating low strain and high strain. When we look at the PTFE exposed surface, as we do shear or sliding shear, we see it's very low levels of strain that are induced. Whereas on the glass, which is the higher coefficient of friction, you get high levels of shear strain. And the take-home message here is that if we mechanically stimulate the PTFE exposed side, we see better accumulation of collagen type 2 and aggrecan, which is what you want in cartilage tissue engineering. So we can control the compressive properties of hydrogels based on increasing the concentration, increasing the photocross-linking time, but we're still not going to get to that state of matching the properties of articulate cartilage. Is there some other way that we can use to get to that stage while maintaining the good environment for the cells? And so we've been investigating over the past several years combining melt-electro-written scaffolds. These are very fine fiber scaffolds on the order of a micron, say 1 to 10 microns, that are 3D printed, and incorporating them into the hydrogels. And the initial study by Visser et al. In Nature Communications showed that you can increase the mechanical stiffness with this reinforcement method by over 50 times by incorporating just a small amount of volume fraction of scaffold. And we've further investigated this, showing that you can actually match the properties of articulate cartilage, both from the dynamic, the static point of view, as well as the dynamic point of view. And it's a versatile approach that can be used with different hydrogels. The real advantage, I think, in addition to that mechanical property advantage, is that the cells are still exposed to that hydrogel matrix, that really three-dimensional environment that is good for their production of new extracellular matrix, rather than being just exposed to a large polymer scaffold, for example, where you might have similar stiffness, but you don't have that three-dimensional environment. Okay, and then we've further gone on to develop zonal structures. By varying the spacing of the fibers, you can get tissues with different mechanical properties. And so with this 3D printing approach, you can generate zonal structures, and this is more similar to what is occurring in the natural cartilage. And then looking at imaging, second harmonic imaging of the tissues that are formed, you can see aligned collagen that's developing around the chondrocytes. So just to wrap up, GELMA is compatible with chondrocytes. It forms a good base ECM for chondrocyte culture, but addition of components like hyaluronic acid or chondroitin sulfate can really help in terms of that cartilage formation. The cells are responsive to mechanical load in vitro as well. Keep in mind, if you're making these gels for applications where you want a smooth, low friction surface, that the substrate will affect the surface properties. And if you want to generate really stiff constructs that are similar to load-bearing tissues like cartilage, you can try this approach where you reinforce the hydrogel with microfibers and match those properties of cartilage. So with that, I'd just like to thank all the people that have worked on this project, people in my group, as well as Dietmar and Owner Boss, funding from ARC. And thanks to Gelomics as well for the opportunity, and happy to take some questions.
Thank you so much, first, for a fantastic presentation. Really, really nice data and some, very well spoken as well. So, got a number of questions here. So I'll just go through them one by one, if you don't mind, and I'll have a few as well myself. So we've got the first question is, sorry, just lost it. Is GELMA suitable for bone tissue engineering? And I guess the concern is around Young's modulus of bone is much higher than that of GELMA. And in that case, what kind of approach can we use to make it more suitable for bone tissue engineering purposes?
Yeah, that's an interesting question. I would say it is suitable for bone tissue engineering. I don't think you want to aim for super stiff gel at the beginning, because you may want to go through a cartilage intermediate phase for one. So starting with a stiffness that's similar to what we're using for cartilage, I think is a reasonable approach. If you do want to have a very stiff construct, like in the cartilage case, I think combining it with a printed scaffold is a good way to do that. Typically, when constructs like this are implanted for bone applications, anyway, you would have a plate or some other mechanical reinforcement as well, so to protect that construct as it's growing.
Thank you, Travis. I think just as a little add-on, I think we also need to differentiate between load-bearing bones and other bones that are non-load-bearing. So potentially, I don't know, for a skull or something like that, where there's not expected to be high mechanical loading environment, it could be useful just by itself. Okay, the next question is, it is very interesting to see that you could control the migration of cells to the gel surface in 3D cell culture. What are the parameters we can optimize to maintain uniform cell distribution in the 3D culture for a longer time?
Yeah, okay, that's an interesting one. We haven't really controlled the migration of cells to the surface. That's something that just happens. We use a pretty high density of cells. It's important to mix your cells well into the gel precursor. But we're using around 10 million cells per mil, which means there are going to be quite a number of cells that are close to the surface. And so those cells near the surface are probably the ones that are coming out. And then once they're on the surface, they can proliferate. Yeah, I don't think we're exactly controlling the migration out of the gel. That's just something that happens.
That's right. Okay, next question. I'm very new to GELMA and hydrogel matrices. Have this been used for establishing airway models? In particular, curious if GELMA can be used for lung-on-a-chip models, providing porous separation between an airway epithelial cells and vascular endothelial cells.
Yeah, I mean, it's not exactly my area, but I think it's a good possibility to establish. I haven't done any research in that area, but GELMA is quite versatile, as I say. You can consider adding things to the system. If there are particular components that are helpful for the epithelial cells, for example, you may want to modify the GELMA system, like we did for cartilage, where we added hyaluronic acid, for example. But certainly it's suitable in terms of providing that three-dimensional framework for cells. Yeah, and you can vary the stiffness as well, which is important. You'd want to go for a softer gel at that if you're going for that.
Thanks. Maybe I'll just add on a couple of thoughts for this question as well. So we should check out our website, because we do have an application that is kind of going along the question of your project aims as well. So we have indeed used GELMA on microfluidic chips to establish microvascular structures, which spontaneously form by the co-culture of endothelial cells with pericytes or smooth muscle cells. that help to reinforce that structure that is building just by vasculogenesis. And we were actually able to, once we applied perfusion to this microfluidic chip system, to create perfusable microcapillaries that are literally just built by the cells themselves. So there's a little bit of like data on our website, under technology you should be able to find that, and I'm happy to answer any more questions later on as well. So we've got one more question here, and then I'll have one or two as well myself. So using infrared laser does not affect the gel to use as the treatment is induced. So I guess the question is whether infrared laser will degrade the gel or interfere with the gel structure in any form.
Okay, that's something, again, I'm not sure. The infrared, we haven't gone into using infrared at all. I mean, I know infrared travels through tissues relatively well. I wouldn't expect it to influence the structure of the gel, but I haven't looked into that.
Yeah, from my experience, I would agree. Infrared is fairly low energy, so it's fairly safe for tissues, and therefore translating this over to the hydrogels, I think Gelma itself forms a very stable gel if it's in the absence of cells that degraded. I think I wouldn't expect any effects on the mechanical properties or the gel properties just by infrared laser treatment.
Yeah.
Okay, we've got one more question. Do you see any negative impact of adding the HA to the Gelma? So I guess this is in relation to the cartilage tissue engineering projects you were talking about.
I don't think there's anything negative that we've seen. The only negative thing, I guess, is it makes it just a little bit more complex. You have to mix in an additional component, but that the effects of the HA have been positive in terms of cartilage, as far as I've seen, and in terms of the mechanical properties.
Absolutely. Okay, Travis, maybe just one last question from my side. I mean, look, cartilage tissue engineering, I think initially has been thought as one of the more easier to get tissues and to be able to engineer something. We only have one cell type. There's no vasculature in it. There's no nerve cells or nerves in it. And really, it's mostly collagen type 2. But then I guess over the years and decades, researchers found out that, well, actually, it's not that easy to create something that has a mechanical functionality like cartilage. And really, it's still an elusive goal that we are trying to aim towards, to be able to one day really generate functional, higher lean cartilage in an in vivo environment. So today you showed us a number of different strategies how to reinforce the hydrogels to make them mechanically, at least from a compressive perspective, very similar to native cartilage. And we also have seen that we can really reduce the frictional properties, which is one of the things that people have largely neglected over the years, and it's getting more and more, I guess, a hot topic now when now that there's a couple of publications that showed how to reinforce gels. But from your perspective, is the Gelma HA mixture a clinically relevant option, or what do we have to change to one day be able to bring something like this into the clinic?
Yeah, I think it is potentially clinically applicable. It obviously needs to be proven in that sense. There is work along those lines, but really improving the mechanical properties through a reinforcement method, I think is quite important. A lot of the hydrogel-based cartilage repair approaches fail because the gel is either not strong enough or is not integrated enough to actually survive that harsh environment. So if we can overcome that hurdle, I think Gelma HA combination is certainly a possible solution to cartilage tissue engineering.
Hey, thank you, Travis. Thank you again, Koen, for your fantastic presentation. Thank you, Travis, for your presentation. It was really great to have you guys here today and present. I'm sure that all of our attendees enjoyed this probably just as much as I did. I really appreciate your time today, and also everyone who joined our webinar and watched us today. So the webinar will also be uploaded on YouTube. So if you want to watch it again later on, it'll all be online and available for you. And before I close the session today, just also wanted to point out, we heard a lot about GELMA and gelatin-derived products. Obviously, Gelomics has a lot of different products in that range, and we are just about to start a new promotion. Actually, we have new GELMA products. So we have bovine-derived GELMA, which is really low in endotoxin. It's a bone-based material, so really good for any type of bone tissue engineering, or where you're interested in looking in cellular biology of bone cells, for example. We've got a porcine material, which is, I think, the standard material, but we'll also be adding very shortly fish-derived gelatin, or GELMA, to our product portfolio, which I think is quite fantastic because it doesn't have that temperature dependency, and it's quite easy to work with at room temperature. So I think that's a nice little spin there as well. So please make sure to check out our website, see the technology tab, where we have an overview of all the different applications of our products and GELMA as well, and make sure to keep an eye out for what's to come in future. So with that, I would just like to say thank you again to everyone. Really appreciate your time today, and hope you enjoyed the session, and we'll see you again very soon.
Thanks.
Thank you.